My project is called Dr. Gene Budger, and it's a web application created to predict modulation of specific genes. And the goal of our project is to budge genes and to budge their expression in a certain direction. Studies in research often find that certain genes, when they're aberrantly expressed, either really overexpressed or knocked down, that can often cause disease. And a big goal in biology and medicine, obviously, is to manipulate these genes' expression to improve the disease state. Our tool is a preliminary technique to identify small molecules that can target expression of single genes. A lot of tools exist now to look at gene sets and look at expression patterns amongst several of them, but what makes our application different is that we can look at single genes. And it's available here. So we want to develop a web application to find small molecules that can maximally influence the expression and empower investigators to enter in a gene symbol and whether they want upregulate or downregulate its expression. And the result is a ranked list of small molecules that can produce that desired expression effect. We start off with two data sets, L1000. That's a technology that assays 1,000 genes in the genome and uses an algorithm to impute the expression of the whole genome. We have 196,000 drug signatures from this data set. CREDS is a crowdsourced extracted data from the gene expression omnibus, and we have 240 drug signatures from there. These signatures were applied to the whole genome, and the results were processed through Lemma, which is an algorithm to determine a modulated p-value. We had a whole bunch of metadata that we got from that database, filtered them by p-value. I only included significant interactions. I made a cutoff arbitrarily at p is 0.01, and then that got fed into a master table. And then you can enter a gene, and then you get a drug list. The way it works is that we have a signatures table and a results table. And each signature has many results. So one drug applied to many genes. Oh, and also we have a fold change and a p-value. Fold change meaning it's been increased 10-fold, 100-fold, and p-value is a significant score. So this is the number of associations per drug. That number can range between 0 and 22,000, which is the whole genome. And so for L1000, the mode is around a few hundred, like 100 to 300, and for the gene expression omnibus, it's about 1,000 per drug. Down here is kind of peculiar because it pretty much affects the whole genome. I'm actually pretty curious to see which kind of drugs those are. So this is a web application. You enter in a gene symbol, whether or not you want to upregulate or downregulate its expression, and pick a data set. You click this button, and the result is a table like this. You might see a drug name several times, but that's just because they're from different geo experiments. So there are different instances and they have different p-values. So we have relevant links to the gene expression omnibus, drug bank, PubChem, drug bank and PubChem if they're actually available, and L1000 data looks similar to this. You can rank these by p-value, a fold change, pretty much anything, and the rows are colored by their fold change value. Darker rows indicate a higher fold change, so I guess if an investigator was to look at this, they would sort by p-value like I've done here, and note, oh, this estradiol has a pretty high fold change and a pretty low p-value. It might be worth checking out. So it's used for preliminary screens for drugs. Reading the CREDS that says fast, but the L1000 is much slower just because its orders of magnitude larger. CREDS has 1 million rows in the table, and L1000 has about 200 million. And the results are useful for perturbing a specific gene, but it's not immediately evident how the drugs will affect the rest of the genome. If you go back to this figure, obviously, if you find some results here, oh, they'll affect your gene, but they'll also affect every other single gene. So that's something to look out for. Nonetheless, we still have great promise because it can still find relevant drugs. So I queried this against several tumor suppressor genes and found some cool findings. BRCA1, which causes breast cancer, if we try to upregulate its expression in order to reverse cancer, we find doxorubicin in our application, and that's an anti-cancer drug that's currently being used. VHL is another type of cancer. Dabrafenib is currently a cancer drug, and This GDC 0879 is actually a RAF inhibitor, and RAF, when it's not expressed in the right way, or when it's overexpressed, cancer can start. And P53, which causes multiple cancers, found homoharringtonine, which is, and triamterene, which are currently being used in clinical trials. So for the future, obviously we want to have more databases. The more we can increase the scope of our project, the better. Better integrate our data sets with experimental metadata. Our table is kind of shoddy in terms of linking to what kind of cells were used and other important information. This really interests me. These tables are really correlative. It would be really cool to see the sort of links between transcription factors and signaling pathways that cause the drugs to cause a desired pattern in expression. So I'd like to thank Zichen Wang and Avi Mayan for invaluable mentorship.